It's really wonderful to see some of your faces, and I look forward to seeing many of you over the course of the next hour and a half or so. I'd like us to begin by introducing one of our community teaching assistants who has been with me now for all 3 runs through medical neuroscience. Her name is Ellen, and Ellen is in the Netherlands, and Ellen has done us all just a tremendous service of building some great resources for medical neuroscience on a wiki page, and I wanted Ellen to just take a couple of minutes and make sure that everyone was aware of that wiki page, and perhaps we can screen share and she can lead us through some of that content. So Ellen, if you would please, I'd love for you to be on and tell us about the wiki for medical neuroscience. Ellen, I think you're muted.
This works better, I think.
There you go.
My microphone. I've made a lot of resources available on the course wiki to help people to find resources and to stop people from spending an enormous amount of time finding good resources on the internet. I want to show it to you how it works. If you go to the like this, do you see it now? Page of the course?
Yes, looks great.
Oh,
We see it.
well, over here you can go to the. If I click that, I should go there. Ooh, he takes him a little. Okay, and then you go to the wiki. First, you have to log in again, and then it says Medical Neuro 003. He just jumped there because I've been there before. This is the main page. Here you see study tips lining up. I will be filling them all the time. In the wiki content, at first I want to go to the virtual lab. In the virtual lab, you have all the units of the course and all the relevant resources there. I want to show you something of Unit 1. So I go there, and to the general resources, I want to just give you an example. You can go to I can't find it at the moment. Oh, this one, The Human Brain by Anatomy Amsterdam. I want to show you something. You go to cerebrum slices, and if you go to the coronal slice, you just can look at all the different slices. That's very useful if you don't have Sylvius, of course, and even if you have Sylvius, it can be useful.
And those are really beautiful brain slices. Those are just an outstanding collection.
And if you put on labels, it gets very busy, but you can still find all the structures that you really need. So now I'm going back to the, oh, I didn't want to do that. I want to go back to the wiki, but that doesn't work somehow. Maybe I can do it better this way. I'm sorry about that, but I'm just trying to find my way in that.
Beautiful brain slices.
I'm back to the virtual lab. And for example, in Unit 2, you have the self-paced neurobiology class from Saylor. And they have all the right resources, and there's people there looking after that all the links and everything are all right. I'm a bit hesitant to start opening that because then I can't find my way back to the wiki, but I advise people who are not very certain about their knowledge on neural signaling to go there and have a look. Another one, very general, is physiology, interactive biology, with very good short tutorials which could give a lot of additional information. You also have a lot of links to animations from the textbook. And I want to go back to the main page of the wiki. And you see that we have other pages. I want to show you neurological disorders. just for example. And you see, well, I'm not I haven't studied medicine or anything at all, so it's difficult for me to think of what clonus looks like. So I have all sorts of examples from that. If ever you want to be motivated again, I advise you to go and see the video of the cochlear implant success story. That will motivate you again to start working, because that's about a girl that was well, lost her hearing at the age of 19 months, and now she had a cochlear implant and she's doing an academic study. And that's very nice material. I also want to show you from the Wiki content resources created and suggested by TAs. That was really what we started with. And I want to show you the brain section cards that B.C. Jackson, one of our TAs, made for you to use. You can use them there from their open source material, so you can use them. You can even use them on work that you share with others. You don't have to be well, there are no problems with copyrights anywhere. And that's really what I wanted to tell you about the course Wiki. We also have the book recommendations, and here you have if you go here, you go to the lines with the study tips, that are the threads that are on the forum that can really, I hope, can help you. Okay. Now I want to go back to the Hangout and choose my, well, that doesn't work anymore, but that was what I was going to tell you about the course Wiki.
Well, thank you very much, Ellen. This course Wiki is so comprehensive and so complete, it makes me feel like I should just quit and not even offer medical neuroscience, just let the Wiki take the place of the course. But really, there's so much that you can learn by going into the Wiki, so I really would encourage everyone, as you progress on through the course, maybe start with the study tips that are associated with each unit, and that would be a great way to enter the Wiki, and then from there you can just discover so many wonderful resources. So thank you, Ellen, and several of the other community teaching assistants that have helped compile all that information.
Well, thank you very much, but I don't agree. I don't think there's anything as important as your tutorials, but that's my opinion.
Well, thank you very much. So I think we'll start taking some questions now. And Shuhong, you've been very patient, and you are one of our first students in the Hangout. So why don't you go ahead and unmute yourself and ask a question?
Yes, sir. Can you hear me?
Yes, I can.
At first, I want to say that I'm very much happy to be in this Hangout with you. I was very much fascinated with your lectures. That was very much comprehensive. And first of all, when I first started this, I was not sure that I will continue or not. But when I go through the lectures, I came to know the very complexity of the brain, and so I was just amazed about it. Now I'm continuing it, and I'm now in the motor system. But some things were so much difficult, and all were not clear to me. Now the mirror motor neuron, which I just read or watched in your videos, it was very much complex, and I couldn't understand how it works. And if you just describe it to me this live, I'll very much happy. Thank you.
Sure. Thank you for the question. So mirror motor neurons were discovered about 15 years ago, and these are neurons that have been found in the premotor part of our motor cortex. And we've also found neurons with similar properties elsewhere in the brain, including in the parietal lobe. But I'll speak about those that are part of the premotor cortex. What was discovered is that there are neurons that are involved in helping to coordinate a particular action. For example, if I were to reach out and pick up an object towards the camera, those neurons were preparing my body for that action, and indeed they were encoding the intention of reaching to grab an object in front of me. Well, what was discovered is that those neurons will fire in a very similar fashion when I observe someone performing the same action, even though my own body is not moving in that way. So I am reaching out to the camera in the hopes that while you observe my hand reaching to the camera, your own mirror motor neurons are firing in your premotor cortex as if your hand is reaching out towards the camera. So Shuhong, I'm going to ask you to do that, and let's see if we can reciprocate that. So reach out towards your webcam with your hand. Go ahead, humor me. There you go. Okay, so my motor system was just encoding that action that I observed in you. That's the essence of the mirror motor neuron system. While you observe my hand reaching to the camera, your own mirror. So I hope that helps you. Is there any other question you'd like to ask?
Yes, sir. I had a question that how memory stores in our brain. Is it like the system that we store memories in the SD card or some other flash memories? Is it like that? Hello?
Well, we still know very little about how memories are actually stored. We do know that as we make a new memory of this moment, for example, while we're having this conversation, that synapses are firing and changing their synaptic weight in the hippocampus. And that is leading to a change in the way activity is now projecting back from the medial temporal lobe to your visual cortex, your auditory cortex, your somatic sensory cortex, all the other brain areas that are processing the sensory experience of this moment. And we think that probably synaptic weights are changing there as well. Now, what we know very little about is how is this information distributed, how is it coded in a distributed and presumably sparse network, and then how is that information brought together again when we recall a memory. But we know that the medial temporal lobe is very important in these functions. So the hippocampal formation and the related cortex in the parahippocampal gyrus is all critical for these processes to work. And something like long-term potentiation is going to be critical for the synaptic plasticity that is necessary to build a memory. But ultimately, there must be some physiological change of the function of the synapses and perhaps even a structural change in the organization of the circuits in all of these different brain areas. Okay, well, thank you very much for the question, and thank you for being with us in medical neuroscience.
Len, I just would like to introduce a couple more people. Welcome everybody who just joined. I see that we have Can and Leonardo and I'm sorry if I pronounced your name wrong, Kayeo. We can just ask to make sure your mics are muted so we don't get any feedback, and when you're ready to ask a question, unmute your mic, ask your question, and then mute it so we can, we don't get any feedback, and then if you have a follow-up. So, all right, thank you.
Why don't we go to Emily next? Emily, would you like to unmute yourself and ask a question?
Hello. Yes, I would like to. I have two questions. The first one is, what is the next step after we finish the course? I found the course very interesting, and I thank you very much for taking the time to make a lot of effort. I can see you must have spent a lot of time doing this course, preparing and giving us the videos. But I would like to know, what can we do after that?
Okay, thank you for the question. There are several things I might say. One is that we are offering a specialization in neuroscience among our course and 2 other courses that are offered from Duke University. If you are interested in the general theme of perception and action and how this relates to the brain and brain function, you can find out more about that on the Coursera website if you look at specializations and then find our specialization called Perception, Action in the Brain. So you could take a couple of other neuroscience courses offered by my colleagues at Duke, and then if you'd like, you can advance to a project that will allow you to apply some of your knowledge and hopefully produce something that is interesting and meaningful to you. So that's one idea. Another idea is to simply go to the Coursera search engine and type brain or neuroscience, and I think you'll discover about 15 or so other courses that might be of interest to you. And then lastly, I'll mention a terrific new online course available from a colleague of mine at Harvard. It's called Fundamentals of Neuroscience, and this is just a fabulous opportunity to learn about neuroscience at a level that's well beyond what I'm able to do. So, for example, there are beautiful graphics and animations and interviews and videos and lots of additional features that are beyond what I can provide for you. So if you're interested in just hearing again about the foundational content of the field of neuroscience, I would send you to that course offered at Harvard. It's called Fundamentals of Neuroscience. So those are just a few ideas about what you can do, and I very much appreciate your interest in medical neuroscience and your participation here. So you mentioned there was a second question, Emily?
It's a very simple question. I was wondering if you could recommend any model, because I struggle to find it, because on a computer you're on a two-dimension only, and I would love to have a 3D brain or something I can play with to better understand how it all connect to each other.
Sure. Well, the best model you could have in your hands, I think, is a model that you yourself make. But if you are looking to purchase a model, you can find models for sale. Unfortunately, they're quite expensive. Usually the cheaper models are about $100 US, and they can go up as high as $1,000 US. So I think that again, you might be just as happy with your learning if you were to find a way to make a model of your own. But otherwise, you can find various commercial suppliers. I'm not quite sure where you are, but you can, I'm sure, purchase something on the internet that you might find very helpful in your learning. Okay, well, thank you, Emily. I think let's see. Tatiana has been here for a while. Would you like to ask a question, Tatiana?
Hello, everyone. I am at the beginning of the course, Unit 1 yet, but, well, my question is a bit abstract. The concepts of consciousness and unconsciousness are well established now?
I would say they are not well established now. We still know very little about how consciousness works in the brain. But did you have more to your question?
No.
Okay. Well, I think we have obviously come a long way in neuroscience, but it's useful to keep in mind that the field of neuroscience really is only about 150 years old. And consciousness and its relationship to the human brain is one of the greatest challenges that we face in life and in neuroscience in particular. So I think that we are a long ways from having an adequate description, except to say that, of course, we believe that consciousness is rooted in the biology of the brain, but we don't fully understand how consciousness arises from nerve cell activity. I personally consider it to be an emergent property of the brain. That is a property that would be very difficult to reduce down to the operations of individual brain cells and individual sets of synaptic connections, although I think we know enough to believe that such operations are essential for there to be conscious brain activity. So obviously this is a very challenging question. We could get into an interesting discussion about the meaning of consciousness in non-humans, or how brain injury or brain damage can alter the nature of human consciousness. These are all very fascinating and very important questions, but unfortunately we don't have an adequate description of brain function that I think will allow us to really address these questions definitively. So perhaps some of you will be inspired to go on to careers in neuroscience and contribute in important ways to this challenge. But for the time being, we'll do the best we can, but appreciate how little we actually do know.
Sorry to interrupt, man. Deborah, can I just ask if your mic is on? Deborah?
Deborah, are you there? Can you hear us?
All right, we're just trying to find out if your microphone is muted or not. We're getting some feedback, so.
Okay, well maybe we can go on to Eric.
Hi, Dr. White. First of all, I want to say thank you for offering this course on Coursera. Although I'm a little behind, I've really enjoyed the lectures I've seen so far. I have two questions. I had one quick question. First of all, my interest is in the development of neurotech, so I was wondering if there are any courses offered through Coursera or other sites that you could recommend for learning a little bit more in depth about the applications of neuroimplants and prosthetic devices.
Great question. Unfortunately, Eric, I don't know of any courses off the top of my head. There is a clinically oriented course from the University of California, San Francisco, on clinical neurology. My guess is that there are some topics that come up in that course, but it's not strategically really designed to address the question you're raising. I think it would be wonderful to have such a course. I have a colleague here at Duke who is a pioneer in the world of brain-machine interface, and I've encouraged him to consider developing a course on that topic. And if not him, for sure someone is soon to do it. So just hang in there, and I think you'll find something in the next year or two. Are you working in that industry, Eric, or aspire to?
Well, right now I'm an engineering student at McMaster University. I'm looking at moving into the development of neurotech and potentially doing it for my master's. And I'm doing a bit of development work on the side into physical augmentation, so exoskeleton tech and things of that nature.
Excellent. Well, I wish you all the best. I have family connections in Hamilton, so it's a nice place to be. I appreciate that part of the world. So thank you for your participation in the course and for joining us here.
Thank you very much.
You're welcome. Okay, how about if we go to Can, or Can? Go ahead, please.
Can you hear me?
Yes, I can. Although, as you say that, I can't hear you now. Perhaps you're muted?
You can hear me now?
Yes, I can. Thank you.
My name is John, actually. It's pronounced like that. Greetings, first of all. Thank you for bringing this course to us, available for all enthusiastic students. And I'm really enjoying your course, and I think you're a great lecturer. You're making difficult concepts way much easier, and thank you so much for everything. I have two questions for you today. First of all, my first question is about various concepts that we've been addressing in our lectures. So my first question is about, we've mentioned some telepathic descending pathways about pain modulation. So I've seen some articles about management of chronic pain with selective serotonin reuptake inhibitors. So we've mentioned in our lectures about some descending serotonergic pathway that modulates activity of the dorsal root neurons. So can you tell a bit about are SSRIs good alternatives, or are they neuroscientifically possible to be used as decreased pain perception?
Yes, thank you for the question. SSRIs, or selective serotonin reuptake inhibitors, might have several important contributions to pain management. One is a possible activity right in the dorsal horn itself. There are projections from serotonergic neurons in the medulla down to the spinal cord that synapse among the complex circuitry of the dorsal horn. Among those local circuit neurons are those that release enkephalins and other kinds of peptide neurotransmitters that can modulate the afferent nociceptive input right at the first synaptic connection in the dorsal horn. So potentially there is a role for serotonin in mediating analgesia right there in the dorsal horn. Now, of course, speaking of serotonin, we understand the serotonergic system much better, actually, up in the forebrain, where serotonin seems to be associated with positive mood states, or at least with being able to shift one's mood state in an appropriate manner. So SSRIs can be very beneficial to people dealing with affective illness. Now, one context in which affective illness is relevant for chronic pain is that many individuals in chronic pain also are at risk for depression. And the SSRIs can help a person cope with the chronic pain by facilitating a more normal, more healthy mood state and affect. So the SSRIs can have both kinds of impacts on being beneficial for what we call the secondary affect associated with chronic pain, and also perhaps with an analgesic effect directly in the dorsal horn. So I hope that's helpful. I wish I knew more about how these systems interact, but I think it's a fascinating topic, and for sure there's more work that needs to be done so that we can better understand how to help people live with pain.
All right, thank you so much for your answer. I have another question about synaptic plasticity, and okay. My question is about what alters synaptic plasticity. So what increases or what decreases the ability of new synapses to potentiate or to be depressed?
Sure. Well, there is a phenomenon that we sometimes call metaplasticity, or the plasticity of plasticity. And I believe that's perhaps what you're asking about. So there are various models that have led to some ideas about how this might work in the brain. Some of them operate on shorter time scales, where it seems to be that the process of plasticity tends to be self-reinforcing. And the associative nature of long-term potentiation can become reinforced and more widespread, suggesting that neurons that are receiving inputs that are active at roughly the same time can be strengthened together as a larger set of the total inputs to a neuron. Now, there may also be plasticity of plasticity that operates over a larger time scale. We call that homeostatic plasticity, with the idea being that there may be some kind of set point within the activity of neurons or even within an entire circuit. And as there may be plasticity at a small scale, these effects are accruing over larger and larger scales, and a system might tend to drift back towards its homeostatic set point, measured either in terms of the total numbers of connections or some integrated average activity pattern over some extended period of time. So there's still a lot we don't understand about how all this works, but I think that the upshot is that the nervous circuits are being tuned to their both recent experience, but also their more protracted history over some period of time.
Thank you so much. That was really explanatory. It was a pleasure meeting you in webcam, and thank you so much for your answer.
Okay, well, thank you for your kind words and your participation in the course. Okay, so we've got a couple of new folks who've joined us, but before we go on to someone new, how about, I'm sorry, you'll have to help me with your name. Is it Kiao? Chiao?
It's Kyle. Can you hear me?
Kyle, yes, we can. Thank you. Welcome.
Okay. Thank you. And first of all, sorry for being late. But the question is kind of, other day I heard a lecture from a professor at MIT who basically said that the computers think, or that they might think, since the form of computer processing information is very similar to the brain. Do you agree with this affirmation? Do you think neuroscience can disprove this statement? What do you think?
Oh, very interesting question. I think that there are powerful metaphors that we can extend to better understand the brain in terms of neural processing that is very similar to what might happen on a silicon chip. So there is a sense in which the brain operates in a manner that is like a computer, particularly with respect to parallel processing. And the converse is also true. I think there are certain designs to artificial neural networks that are inspired, at least, by our understanding of the circuitry of the human brain. Now, with that said, I do think there are limits. I think that the brain is an embodied organ, and it is an organ that exists within the context of the human form, if we're talking about ourselves, or what other kind of organism that we may have in mind. And so while it's tempting to think of the brain as a set of circuits that can be understood in isolation of the rest of the body, I actually believe that in order to fully understand the brain, we need to appreciate it as an embodied organ and understand how the brain interacts with the rest of the physiology and the rest of the anatomy of the organism. Now, with that said, I am excited about the future. I think there will be some really interesting challenges, both ethically, morally, biologically, as computer science continues to advance and we see the development of artificial intelligence. I just had this discussion in a class that I'm teaching at Duke last week, which is really focused on the notion of movement. And we are reading some very interesting essays from a philosopher named Sheets Johnstone, who argues that movement is knowledge. And if that is the case, then one might wonder whether there is a kind of robotic intelligence that can be embodied by patterns of movements that non-animate machines are capable of producing. So I'm not sure about where that might go, but I do think there are interesting dialogues between computer scientists and brain scientists and bioethicists that are really challenging our notions of what is sentience and what is consciousness. So these are great questions. I wish I had something more authoritative or definitive to say, but thank you for raising the issue.
Thank you.
Okay, how about if we go to Natalie next? Natalie, are you there? Natalie, you might be muted if you are there.
Natalie, do you have a camera? Okay.
Well, we'll try to get back to Natalie. How about Talay?
Talay, can you hear me? I think that's strike two.
Okay. Well, perhaps someone who's already been on and asked a question would like to speak up and contribute with another question or comment.
I'd like to ask, what you think, it's about ethics. What do you think about deleting a memory of a person or of someone?
Could you phrase your question again, please? I'm not quite sure what you're asking about a memory.
Okay. My question is, what's your opinion about erasing a certain kind of memory of somebody?
Yeah. Well, fascinating question. I think that we can have a faulty memory, right? That can happen just because memory is not 100% efficient. We can have a brain injury that can damage our ability to make new memory. Or we can have an acute injury and have what we call retrograde amnesia, which is a loss of memories that were essentially in process before they were fully laid down into long-term memory. I think it would be extremely difficult to selectively erase a memory of a person. Now, there may be psychological factors that affect our ability to recall memories of certain individuals in the context of post-traumatic stress or post-traumatic trauma, for example. But otherwise, longer-term memories are remarkably resilient. And I think the reason for that is that they are stored in a highly distributed fashion. So it's very difficult to eliminate all the distributed pieces that constitute that memory as they are spread out in so many different parts of the brain. So I think that your question is reminding me of a piece of data that was published, I think about a year ago now, where there was a suggestion that maybe we could actually create memories, essentially false memories, by manipulating the activity within the medial temporal lobe circuits of the brain. Now, this was done in an animal model. We are nowhere near being able to manipulate the human brain in a way that would either introduce or erase a particular memory. But in principle, one might think about that possibility. I'm happy that we are a long ways away from understanding how to do that, because I think that's an ethical dilemma that would be very challenging to think through. So I don't know that I've given you a great answer to your question, but hopefully you've had some things to think about there. So what do you think?
I think yeah, it's an ethical thing. I think all the facts that we pass through life, it collaborates somehow for us to learn something and make it better. So I think it's an object of discussion. It's surely is.
Okay, great. Well, thank you for that additional question.
It's the first time that I'm here using Google and Hangout, and I'm a little lost. But I'm loving the course. You're a great teacher, and I'm enjoying every single minute of it. Okay.
Well, thank you very much. I know I'm asking an awful lot of you and everybody else, and I appreciate all of your hard work and your dedication to this subject matter.
Okay.
So, Emily, you're still there. Is there anything else you might want to ask or anything else you might want to say? Perhaps not. So let's see.
I just clicked on the wrong button. No, I mean, I must say I don't know how people find the course. I find it very difficult. I have a full-time job and I do that on top of it, and it's nothing to do. I'm a mathematician, so nothing to do with neuroscience to start with. And I really enjoy your video, but I think I'm lacking the lab practice or whatever you. And I have no clue how I can gain that. That's why I was asking about the model. But I don't know if there's any group of things you know of that could have people like me that are not really into academics anymore. being able to practice or do something.
Sure. Well, I don't know if you were on when Ellen was going through some of the resources on the wiki, but I think you might enjoy browsing some of the anatomical resources that are there that show you some models of the brain. There are at least a couple of links to three-dimensional models of the brain that you might find, and I really appreciate your desire to actually put something in your hands. I think that's very important and very helpful. But if we can't actually get something in your hands, then at least you can interact in a useful way with some of these brain models that might help you appreciate the relation of structure and function in the actual human brain. So that's probably the best thing I can recommend to you, aside from browsing the wonderful peer assignments that have been posted on the discussion forum. I hope you've all had a chance to see some of those. Some of them are just tremendously creative. Some people have made brain models. One individual arranged food on their plate in the shape of the lobes of the cerebral hemispheres, which I found to be just really fascinating and entertaining. So hopefully you can find creative ways to make visible and tangible your knowledge of the brain as you progress through your studies.
One other question is, how do you see I could use my mathematical knowledge in neuroscience? Is there any interaction you can think of?
Sure. I think that a very exciting domain of biology in general, and neuroscience in particular, is computational biology or computational neuroscience. One of the challenges we face in the field is that we have so much information that we often don't really know how it will lead us to greater understanding. And what we really need is theory. And that's where I think mathematics comes in, because I think we need a quantitative approach to understanding the data that are being generated. So one can think about actually applying particular mathematical principles to data sets from neuroscience, or one could think about actually trying to derive quantitative theories of brain function or brain organization. Some of my research that I introduce in Unit 3 of the course pertaining to the organization of circuitry in the visual cortex is designed along these lines, where I've been collaborating with some physicists and some mathematicians to try to understand the organization of circuitry in the brain. And as I briefly explained in one of those videos, one of the surprising results is that we've discovered the mathematical constant pi in the organization of brain circuits. So that was a big surprise. We never would have anticipated that, but I think it demonstrates that the brain, like the rest of the universe, is subject to the physics of the world in which we live, and there can be quantitative descriptions of the interactions that take place to even give rise to circuitry in the cerebral cortex, as our paper showed. So I think there is an urgent need, actually, for mathematicians and computer scientists and theorists to engage with our discovery of neuroscience. Because, as I suggested, I think what we are really lacking is good theory that would guide the empirical research and the interpretation of the data we're collecting. So perhaps you have a future in neuroscience contributing in your discipline. Okay, maybe we can try to get back to Natalie. Natalie, are you there? If you are, could you please unmute your microphone, because we can't hear you or see you. Okay, maybe she's not there. Is there anyone else who'd like to speak up?
Sir,
Yes.
I have a question.
Go ahead.
Sir, you said that the mammalian cycle of locomotion is organized by central petal generators in the spinal cord. Hello,
Yes.
sir. In that portion, you showed a picture of a cat that was cut down in the spinal cord, and it was moving like the previous one. But how this is possible? Though I cut down the spinal cord,
Right.
I couldn't
So,
understand this properly.
right. So the idea is that in order to engage in locomotion, there needs to be coordination of the output of lower motor neurons to the flexor and extensor muscles of the leg, and there needs to be coordination across the body axis from one leg to the next. And in some of the animal experiments that have been done with quadrupedal animals, scientists have asked, can locomotion patterns survive a transection of the spinal cord? And the surprising answer is yes, they can. And this is the first evidence for central pattern generation in the mammalian spinal cord. So the idea is that there are local circuits within the spinal cord that are connecting the motor neurons that innervate flexor and extensor muscles and coordinating the timing of their output. So it's really the coordination of the timing of the output that's critical here. So just think about what has to be done with each step cycle. There has to be flexion of the limb so that the limb can advance to the next contact with the ground. And then once that contact is made, now there needs to be extension as the leg supports the weight of the animal against gravity. So there's this rhythmic cycle of flexion and extension. And then depending upon the pattern of locomotion, either the two limbs on the same side of the body are working together or they're working in antiphase with one another. So there must be local circuits that can coordinate the output that's necessary for the particular pattern of locomotion. Now, you might have thought that, well, doesn't the motor cortex do all of that? Well, the answer is no. The motor cortex provides the signal about the intention to move, to start walking, for example, and it provides an ongoing signal that's necessary to sustain locomotion. But the motor cortex is not coordinating the details of walking or stepping. That's being coordinated at the local level of the spinal cord. Now, when it comes to the human nervous system, things are always more complex than we might imagine. And so one might wonder, well, why don't we see more robust stepping for humans that have suffered spinal cord injury in the way we see it for the cat? And we don't really quite understand the answer to that question, but presumably it has to do with the fact that in the human, the spinal cord seems to be more dependent upon descending input that's coming from higher centers in the brain than in an animal such as the cat. But nevertheless, I think we have reason to believe that we have central pattern generators that are governing these kinds of rhythmical behaviors, such as walking, stepping, swimming, some of the things we do with our craniofacial regions, such as chewing. These are all examples of local circuits coordinating the activity of the muscles that are necessary for rhythmical, stereotypical movement. So we would love to know how we can activate these central pattern generators in individuals with spinal cord injury, and there is quite a bit of investigation aimed at understanding how to do so. So I hope that's helpful. We still depend upon these simple animal models to try to understand this, and there's much more that we need to know to really have those breakthroughs of treatment for people that have had interruptions of the connection between the brain and the spinal cord.
Len, I have kind of not really a question myself, but kind of ties into Emily's last question about being a mathematician. I kind of found it pretty interesting that Lizine McCune, Lisa, she had asked a question about, first she said that she really appreciates the class and that she's actually fell in love with neuroscience and want to find out or want to know if being 27 is too old to be a good neuroscientist.
Sure. So that was Nick sitting next to me here in the room. He's the official host of this Hangout. And Nick, I ask you to mute your microphone. Thank you. So Nick was helping to solicit some questions from some of you all, and he was just reading a question that came into us about, is it too old to be 27 and interested in neuroscience? Of course, my answer is no. It's never too old to be interested in neuroscience and actually want to do something about it. Now, of course, science is for the young and for the young at heart, sometimes for the young of body as well. But I do think if you're interested in neuroscience, there are many ways in which you can apply your learning and your knowledge, even without necessarily becoming a professional neuroscience researcher, as I am. There may be lots of ways for you to apply your knowledge of the brain in a variety of different professions. And we see just tremendously rich dialogue happening among the disciplines, among the professions. So you may be interested in business, you may be interested in law, you may be interested in the arts or the humanities, and regardless of your avocation or your vocation, I think you'll find an opportunity to apply what you're learning, even in medical neuroscience, to those other fields. But if you are thinking about pursuing graduate school in neuroscience, I think it's never too late. You just have to understand the commitment that's necessary to pursue doctoral education in the neuroscience field. And if you're prepared to do that, then I certainly would encourage you to pursue your dreams, whatever they may be, even if they are to become a neuroscientist yourself. So I hope some of you would aspire to that. But I want you to understand that you need not be a professional neuroscientist to actually learn about the brain and apply what you're learning to whatever enterprise you find yourself pursuing in life. In fact, I don't know that we've reported much about who all is with us in medical neuroscience yet, but the distribution of ages is really, really impressive. So we have students from the middle school years, young teenagers, all the way up to folks that are well into their 70s and I suspect even into their 80s that are joining us here in this great journey together in medical neuroscience. So I think our diversity as a learning community is truly our greatest strength. So thank you, all of you, wherever you're coming from in life, whatever your age, whatever your occupation. It's really great to have you all here in this community. Okay, Ima, is that you? Are you ready to join us and unmute your microphone and ask a question or share a comment?
Can you hear me?
Yes, I can. Welcome.
Okay. Hello, Dr. White. I first like to thank you for your amazing course and that you made it available to us through Coursera. I'm actually having a neuroanatomy course in university, and I'm doing your course as a supplement for it, and it's helping me so much, and it's just great.
Thank you. I'm delighted to hear that.
Yeah. Now to my question. My professor in my biochemistry class once mentioned that in general the membrane is made of unsaturated fats, and that these unsaturated fats are part of the building blocks of the membrane, and that this would be really important for the central nervous system. But he didn't elaborate why that would be so important for the central nervous system. I was wondering what the relationship in that is between nervous system and unsaturated fats.
Well, my guess is that he is thinking of the importance of myelin in providing insulation for axons that facilitates the conduction of electrical signals along the length of an axon. Since axons have the special challenge of being able to generate and then propagate an electrical signal over tremendously long distances, unlike any other kind of cells in our body. So the ability to make that propagation, that transfer of charge as efficient as possible, with as much fidelity as possible is extremely important in the nervous system. So my guess is that he is recalling that myelin is essentially a rich layering of membrane, which is essentially a phospholipid bilayer that is essential for insulating the axon, helping to reduce the leak of current, and helping to maximize the passive conduction of electrical charge from one site of action potential generation to the next. So without being in your class, I only imagining what is in the mind of your professor, but that very well may be it.
Yeah, now that you mentioned it, it makes such clear sense, and I wonder why it didn't occur to myself. And I have one more question, if I may add.
The axon, helping to reduce. Yes, go ahead.
And that is it true that in every person one hemisphere is dominant over the other?
Well, I believe there are domains of brain function that are lateralized, and we could identify a dominant hemisphere in most individuals. Now, we would have to be very cautious about really what are we talking about. Are we talking about language? Are we talking about music? Are we talking about spatial attention? So there are different functions that are lateralized to one hemisphere or the other. I can tell you, speaking from a clinical point of view, most clinicians, be they physicians or some other kind of medical professional, think about language as being the anchor for dominance. But that's not always going to be the case. There may be some brains that are simply set up differently, where language is dominant in one hemisphere, but there may be some very compelling aspects of human behavior that may be lateralized to the opposite hemisphere. So if one were to ask which hemisphere is dominant, it would very much depend upon what kind of function you're studying. So just understand that from a clinical standpoint, most medical professionals will have language function in mind. But of course, there's much more to human behavior than language, as important as that is. And the two hemispheres work cooperatively in all manner of human behavior. So that's the other point I would make, is that even when we talk about dominance, we're not talking about an absolute allocation of neural function to one hemisphere or another. Rather, we're talking about a consolidation of circuitry that makes one hemisphere either more active or more critical to the expression of the behavior in question. But you can be sure that the counterparts in the so-called non-dominant hemisphere are also contributing, perhaps just in less obvious ways. So, for example, in language, for most of us, the left hemisphere is what helps us understand language and helps us make language. But the right hemisphere is contributing with imparting our speech with emotion and helping us to interpret the emotion in the language that we receive, be it through our sense of hearing or even our sense of sight and touch. So both hemispheres are involved, but one we might say is dominant because it is responsible for those aspects of human behavior that are easiest for us to assess and appreciate. And that's often what the neurologist or the healthcare professional might be most concerned about. So thanks for the question. It's an interesting topic. We'll get to more of this matter of hemisphere lateralization when we get into Unit 6 of the course. So I don't suspect you're there yet, but when you do get there, you'll learn a little bit more about lateralization at that point. Okay, thank you, Ima. It's nice to meet you, and thank you for your participation in the course. I wish you all the best in your studies of biochemistry and neuroanatomy and other things that you're doing at your own university.
We would like to introduce Anna to the room.
Hello, Anna.
Hello, Professor White. I hope you can hear me.
Yes, I can. Welcome.
Thank you, and thank you for your very nice course. And I'm really excited about being able to participate in this hangout, because to imagine that we are in totally different places at the moment, but still meeting each other and talking about neuroscience, I think that's fascinating.
Yes, indeed.
And I am actually an active researcher in my final year of PhD, or PhD studies. So I have enough of my hands-on experience, although not always or not at all on human brain, but still the knowledge helps me with the animal studies that I'm pursuing. But what I think is very challenging is that every day brings more and more knowledge on single neurons and neural circuits on a very detailed level, especially concerning morphology and physiology. And I think it's very difficult, even for scientists that are very advanced in that field, to piece it all together and make like a bigger picture of that. So I think. Sorry?
Go ahead, Anna. That was
just a thought of mine. But what I very personally think is very interesting is our dreams and thoughts, and how their content is actually encoded by brain activity. And I have read some stuff that some groups are actually working actively on that topic, to like using algorithms to link visual stimuli, I think, to computed neural signals. I'm not sure I recall that correctly, but I was wondering what your take on that would be. Do you think that neuroscience will have an answer to that sometime in the future, like to know why we think what we think and what we dream? And do you think it's rather subject of psychological studies, or maybe something on the border of these fields?
Thank you for your question, Anna, and thank you for your work in neuroscience. I certainly wish you all the best as you complete your doctoral studies and then move on into the career that hopefully is your dream. So I wish you all the best there. What I would say is that I think that we need both psychology and neuroscience to address questions such as the one that you raise. We need to understand the mind as an object of study in and of itself, without being burdened by neurophysiology and neuroanatomy. But in order to have a deep understanding of the mind, we do need to address the underlying neuroanatomy and neurophysiology. So we need both perspectives. Sometimes we have insight that comes from, let's say, the cognitive sciences that can inform neuroscientific investigations in more reduced systems where we do have access to nerve cell activity and the organization of circuits. And vice versa. Sometimes we can study properties in the reduced systems, and that might predict some behavioral consequences, even in functions as interesting and as complex as consciousness, dreams, and imagination. So I think we definitely need both realms of activity to have a more complete model of understanding brain function and how it relates to behavior, even cognition. But I do think we're getting closer. So for example, there are some fascinating studies that you may have heard about where brain cell activity is recorded in awake and behaving animals, and then that same set of microelectrodes is recording activity while animals go through various stages of sleep. And what has been observed in some of these studies is something that looks like a replay of the day's patterns of activity during times of rest and sleep, almost as if one function of sleep is to consolidate the experiences and the memories that were laid down in life during our waking state. And some of these activities seem to be reflective of a role of sleep, not just as a restorative function, but actually as a function that's essential for the consolidation of memory. So I think there are some really fascinating studies that are ongoing, that are beginning to shed new light into some of these matters, and they require both behavioral science and neuroscience, particularly neurophysiology. But an understanding of the circuitry and the organization from an anatomical perspective also is a critical framework that needs to be brought to bear to this work. So just one small example of all of the exciting work that's going on in the field of neuroscience. And you're absolutely right. The knowledge is exploding, and we are discovering more and more every day. And it's a real challenge to make sense of it all and to keep up with the rapid pace of discovery. So that's the world you will find yourself in very shortly if you're not there already. But it's a tremendously exciting time to be interested in neuroscience. We just have to be up for the challenge of keeping pace with the new discovery that's coming upon us every day. So thank you, Anna. Thank you for your participation in the course, and thank you for your question.
Does anybody else have any questions?
Sorry.
Maybe Nick can give us another question that was submitted on our questionnaire that we sent out a few days ago.
I actually was just looking at that. So this one came from Bayu Gautama Wandari. I'm not sure if this is up your alley or anything, but his question is, I'm curious about the application of optogenetics on the human eye to replace the degenerated photoreceptors in the retina. Does this technique have a big potential to give blind people sight like a normal person has, and what are the limitations of this technology in the context of technology for the blind?
Okay, well, thank you for that question. Some of you may know about optogenetics. It is a relatively new technology in neuroscience that's based on the encoding of light-sensitive ion channels in brain cells. So these are ion channels that come from cyanobacteria and other kinds of very, very simple organisms that can be put into nerve cells so that those cells are now sensitive to light. So basically what's happening, very much like the opsin protein in our photoreceptors, a photon will interact with a protein, and that protein has been engineered to gate an ion channel, allowing, for example, sodium ions to rush into a cell, making it an excitatory event, or chloride ions to rush into the cell, which might make it an inhibitory event. So the idea is that perhaps we can put these light-sensitive ion channels into retinal ganglion cells, and that could be an effective intervention for an individual that has had degeneration of the photoreceptors. So rather than trying to salvage the photoreceptor itself, maybe we can make the ganglion cell photosensitive. So I think there is great potential for this to be a very exciting new approach to dealing with certain degenerative conditions that particularly affect the photoreceptor layer of the retina. One of the lessons I think we've learned in this field of neuroprostheses is that we shouldn't worry so much about getting the stimulation of the sensory organ perfect, because I think what we've learned is that the brain can do marvelous things to try to interpret even very, very crude signals. We've learned this, for example, in the history of cochlear implants, where the very first devices were very crude from an engineering perspective and a neurophysiological point of view. Yet, I think we learned that the auditory system can make sense of these signals, and there can be plasticity in the auditory pathway that can lead to enhanced function, even with fairly crude signals being delivered to the sensory organ. My hope is that something like that will happen in vision as well, that we will understand that even fairly crude patterns of stimulation of the neural elements of the retina may be interpreted by the brain in a useful manner. So that's essentially reason to be hopeful in this approach, but for sure we do want to try to engineer these optogenetic methods to increase the amount of information that can be encoded in the retina itself so the brain has more information upon which to operate. So thank you for that question about optogenetics, and let's see if there's someone else who wishes to ask a question that's in the Hangout now.
Sir.
Yes.
I want to make a question.
Yes, go ahead. Shoot.
You said about the synaptic plasticity. In case of synaptic plasticity, the role of calcium is the critical. And
Yes.
if the concentration is larger, then it can upregulate the AMPA receptor. And if the concentration is lower, that will downregulate. Why this concentration is so much critical in this case? I can't understand actually why this is phosphorylation.
Right.
Thank you,
So,
sir.
yes, thank you for the question. So, calcium is really a key intracellular second messenger that helps us understand the difference between strengthening a synapse or weakening the synapse. Now, in the context of the cerebral cortex, if there is a sudden rush of calcium into the postsynaptic process, then second messenger systems will be activated that lead to the phosphorylation of target proteins, and some of those will insert new AMPA receptors into the spine, making that synapse stronger, more responsive to the glutamate that's being released. Now, if the influx of calcium is at a lower level and at a slower rate, then second messenger systems that will dephosphorylate those target proteins are more likely to be activated. So one way to address your question is to say, well, what's the difference between activating a protein kinase and a protein phosphatase? And the difference is that these two different kinds of intracellular systems have different affinities for calcium. Okay, typically the way this works is that the protein phosphatases have a higher affinity for calcium than do the protein kinases. So what this means is that if just a small amount of calcium has entered the postsynaptic process, it's going to be more likely to activate the protein phosphatases because they have a higher affinity for calcium. The kinases might be blind to the presence of that low level of calcium. On the other hand, if there is a sudden rush of calcium and it reaches high concentrations quickly, it is very likely then to interact with the protein kinases, which have a somewhat lower affinity and require a greater concentration. But presumably the balance of phosphatases and kinases is such that with a rapid rise in calcium within a spine, for example, then the kinase system will be activated and that will lead to potentiation. So by varying the affinity of these calcium-sensitive mechanisms, we can bias the operation of plasticity towards potentiation or depression. And I should just add that I was speaking of the context of the cerebral cortex, and the cerebellum things are a bit different because there the increase in calcium that we see, that we associate with plasticity, actually promotes the internalization of AMPA receptors. So there, the dominant form of plasticity in the Purkinje cell is long-term depression rather than long-term potentiation. And the difference between the cerebellar cortex and the cerebral cortex is the kind of second messenger systems that are activated by a sudden rush of calcium. You know, calcium is really fascinating in so many respects. One additional aspect I should mention is that if the influx of calcium is to very, very high levels, that of course can be dangerous, and it can lead to the destruction of the cell by the activation of excitotoxicity and various second messenger systems that can lead to changes that will actually destroy the neuron. So calcium is essential for plasticity, but it can actually lead to the death of the neuron if the concentration should rise too rapidly to too great an extent. So there are multiple mechanisms within the cells that are buffering calcium as well. That's the other side of the story, is that whatever the change in calcium homeostasis, we want it to be controlled and we want it to be short-lived so that it can be tied to the events that might lead to a change in synaptic weight without overwhelming the cell, possibly leading to its own destruction. So thank you for that interesting question, Shuhong.
Thank you very much, sir.
Let's see if anyone else would like to ask another question before we move on to another submitted one.
Can I?
Yeah, go right ahead.
Well, my question is not too much a technical one, but is a more general. Do you think that neuroscience would be able to prove or to disprove if we do have or if we don't have free will? Because nowadays, a possible answer I once heard would be to say that we do have free will, as it was my brain that activated such a neuron and other things and made my arm move, for example, and etc. But I still don't think that's a good answer, and the problem still stands. So again, do you think that neuroscience can prove or disprove if you do have or don't have free will, and why?
I'm going to give you the easy answer, and my easy answer is no. I don't think that neuroscience will allow us to prove or disprove the notion of free will. I think it will inform the debate, and it might even change the debate. But I do not believe that neuroscience will answer the question. And partly I say that because we still know so little about the nature of human consciousness and the complex neurobiology of decision making. I think we will get closer to that point over the coming decades, and for sure in the coming century. But I really have my doubts as to whether we will understand the brain deeply enough to address questions that really have a core philosophical basis to them. So I think neuroscience will never replace philosophy. That's my own biased view. But I think it can inform philosophy, and I think it goes the other way around as well. I think neuroscientists can learn a lot by engaging with philosophers, and we might design better experiments that lead to greater insight about the underlying brain mechanisms. I know there are some folks out there who have stated in the past that philosophy, psychology, cognitive science will all reduce to neuroscience. I don't particularly believe that. I think that we need these other disciplines, and that there are properties, I think, of the human mind that are emergent and will just always be beyond the reach of the brain scientist to provide a complete, comprehensive description of the underlying phenomenon. So I'd love to be wrong about that. Maybe time will tell. But my current sense of this field is that some of the challenges of human consciousness, including free will, are just beyond, I think, the tools in reach of neuroscience. What do you think about this question? Surely you must have an opinion.
Thank you very much. Yes, I do have. I am a philosophy student as well as a psychology student, and I agree with you. I don't think we'll be able to tell if we don't do or if we don't have free will. There are certain questions that I think we won't be able to answer, such as free will, like existence of God, etc. So I agree with you, but I don't know.
Okay. Well, thank you so much. I appreciate your being with us and for asking such a challenging and interesting question. Folks, I think I'm going to have to sign off here. I do have another appointment in my day that is about to happen shortly, and I am having just a bit of a problem with my back today, so I think I need to change my position and my posture here. But thank you all so much for your participation with us in this Hangout, and thank you for all of your hard work in medical neuroscience. It's just so fantastic to have a chance to see many of you and to speak with you, even in this medium. And I just thank you for your interest in the neuroscience field generally. And I really look forward to what each one of you will do with this new knowledge you're acquiring. So I'll just encourage you to hang with us. We're just getting started in medical neuroscience. We've got another 5, 6, 7 weeks to go, depending on where you are in your studies. And I look forward to the rest of the journey with you all. So thank you for being with us. And with that, I think I'll ask Nick to conclude our Hangout. And for those of you watching on the YouTube channel, it will soon be posted there. So if there's something you'd like to review, we can all do so at that time. So thank you all, and I look forward to seeing you in the discussion forum and in our next Hangout, which will be coming up probably in just a few weeks. We'll let you know about that. Okay, take care, everyone.
All right, that concludes our medical neuroscience hangout. I hope everybody enjoyed it, and I hope this is the first of a couple, or hopefully many, for the next, what, 6, 7 weeks, you said?
Something like
that. So hopefully we'll have some more, so stay tuned. All right, bye.